Hi everyone, I'm Elena Losanno, a PhD student at Scuola Superiore Sant'Anna in Pisa, Italy, and I will talk about an implantable brain-body interface using intrafascicular stimulation to restore hand functions. Brain-body interfaces have emerged as a solution to restore voluntary hand control in people with paralysis. And recently, there have been some clinical demonstration of brain-body interfaces, such as the study of Ajiboy and colleagues. However, we are still far from the restoration of the full hand dexterity. The difficulty is that we need to tackle two different models, one for motor decoding and one for movement restoration and their interaction. Ideally, we would like the motor decoding to be easy to learn, accurate and stable, and the movement restoration to be selective and resistant to fatigue. In this work, we try to tackle all these issues. For the motor decoding, we built on recent findings that our neural activity is constrained in the low-dimensional space called neural manifold, which can shape learning. And we designed a decoding strategy in which the dynamics of intrinsic neural ensembles was directly mapped to the output variables. We tested this strategy, which we call manifold-based direct control, in a monkey, using a cursor control task, and found that this strategy is easy to learn and stable. For the movement restoration, we propose using peripheral nerve stimulation at the intrafascicular level. In 3 monkeys, we found that intrafascicular stimulation can evoke multiple flexion and extension movements of the hand with only 2 implants, and that functional force levels can be produced and sustained over time. Finally, we integrated these 2 modules into a full brain-body interface, and we tested it in a monkey. And we found that the animal could use this approach to voluntarily control smoothed hand opening and closing.